Hello viewers, in my last session I already discussed about structural and functional organization of vertebrate gastrointestinal tract, and now I start discussion about physiology of digestion in vertebrate, and this is very important for NEET students, the students of honors and pass courses of zoology, botany and microbiology, and also for different competitive exam. Physiology of digestion can broadly be categorized into mechanical digestion and chemical digestion. The mechanical digestion includes different mechanical processes. Number one is chewing, which is performed by mouth. The chewing is facilitated by the teeth and tongue and epiglottis also. First, the food taken into the mouth undergoes the grinding and also mastication by teeth, and also breaks down into the smaller particles. And then the tongue pushes the food back towards the throat and pushes it to take its entry into the esophagus in the form of a bolus. When the food, the bolus, enters into the esophagus, then epiglottis prevent bolus from entering it into the trachea, and here also uvula also plays an important role. The next process is churning, and it occurs in the stomach. When the food travels along the esophagus and enters into the stomach, then the stomach lining muscles start squeezing and mixing the food with the different digestive juices secreted from the stomach, and then the food is digested within the stomach for several hours and ultimately turns into a creamy paste which is called chyme, and then the chyme enters into the first part of intestine, that is duodenum. The another mechanical process also involved is the movement of food, which may be of two types, peristalsis and segmentation also. The peristalsis is mainly performed by the esophagus, and also it is performed by the stomach and gut. In esophagus, the adjacent segments of the esophagus, the adjacent segments are these segments, that is this segment, the groove-like structure, and the swollen structure. These two are the adjacent segments, and the adjacent segments, it contains the smooth longitudinal muscles. Their contraction and relaxation helps the forward movement of this, or the unidirectional movement of the food bolus into one direction, and also in the segmentation process, mainly performed by the intestine. Here the non-adjacent segment, these two segments are non-adjacent segment because this is separated by a groove. So these two non-adjacent segment contains the circular muscles, and the circular muscles start contraction and relaxation for the both side movement, towards both directional movement of the food bolus, for a greater mixing of the food with digestive juice, and the overall segmentation process helps the chyme for its bidirectional propulsion, and also to make slower movement of this chyme, the overall slower movement of this chyme. Then I come to the chemical digestion, and first of all we should know that what are the chemical agents which perform the chemical digestion. The chemical agents are the stomach acids, bile, and the enzymes. The stomach acids, the stomach acids, we know that they are present within the stomach, and also these acids, that is digestive acids, also released from different gastric glands of the stomach. And since this is acid, so it maintains a low pH level, that is pH around 2. This acidic environment helps to denature the protein component of the food and the macromolecules, to break it different micromolecules, and also help in overall digestion. The pancreas also release the bicarbonate ions, which is an alkaline compound, which also neutralizes this acid before its entry into the intestine, because in the intestine it will find the neutral environment. Now I come to bile. This bile is produced from liver and also stored in the gallbladder. This bile contains the bile salts which also interact with the fat material, the fat globules of the food material, to divide them into the smaller droplets by the process which is called emulsification. And also different enzymes act as chemical agent. The enzymes are the biological catalyst. We know that it speeds up the rate of the chemical digestion, and the different digestive enzymes secreted by mainly the stomach, pancreas, and also the salivary glands, and they digest most macromolecules into the micromolecules, that is monomers in the small intestine. And generally enzyme acts on the substrate for the hydrolysis of different macromolecules to break it into the different micromolecules. And now we are going to the main part of the procedure of the digestion of carbohydrate. The carbohydrate digestion starts in the mouth with the release of amylase from the salivary gland, and in the small intestine the pancreatic amylase also helps in starch and carbohydrate digestion. Now we go through this flowchart that in the mouth the salivary amylase acts on the starch and glycogen to break it into the disaccharides. Means these disaccharides are maltose, sucrose, and lactose. And this in the intestine, the enzymes, that is maltase, sucrase, lactase, act upon them. The maltase act on maltose to break it into glucose molecules. Sucrase act on sucrose to break it into glucose and fructose molecules. Lactase act on lactose substrate to break it into the glucose and galactose. So in the smaller intestine also, the brass border enzyme, that is the we can call it the alpha dextrinase. This alpha dextrinase also act on alpha dextrin and ultimately breaks it into one glucose unit at a time. And also the cellulase enzyme also act on cellulose, but in human being there is no cellulase, but in herbivore mammals we can find the cellulase enzyme which acts on cellulose. Now I come to digestion of protein. Digestion of protein starts in stomach, where the protease, that is the pepsin and HCl, acts on the protein components of the food and breaks it into the smaller polypeptides. And we go through this flow chart, that is the pepsin acts on protein to break it into large polypeptides. Again it acts on the large polypeptides to break it into short peptides and also amino acids. Now in the small intestine, the brass border enzyme, that is the mainly the endopeptides, which is released from the pancreas, also acts on these peptides to break down it, and this enzyme is endopeptidase. Here also in the intestine, also small intestine also rather, there are also two enzymes, that is chymotrypsin and trypsin, also act on the specific bonds on the amino acid sequence. Again another two enzymes, that is the brass border, which is called brass border enzymes, they act in small intestine. They act on aminopeptidases and dipeptidase. The two enzymes, aminopeptidase and dipeptidase, they act on the polypeptides to further break it into the smaller parts of the peptide chains and ultimately into the amino acid, which last of all enter into the bloodstream. Lastly, I come to, not lastly, but previous part before the last part, that is the digestion of lipid and fat. The lipid breakdown occurs in intestine. You know, in intestine the lipid breakdown occurs it begins with the emulsification of the fat globules. The emulsification of the fat globules by bile, which is released from gallbladder. The smaller fat droplets is again digested by the lipase, and this lipase released from pancreas. And the lipase may also come from different sites, that is, they may be lingual lipase, gastric lipase, pancreatic lipase. These three lipases responsible for the lipid digestion, and the gallbladder release bile, that is, this bile contains the bile salts, which is again composed of 4 different kinds of free bile acids. This is a cholic, deoxycholic, quinodeoxycholic acids, and also lithocholic acids. Each of these acids may in turn they combine to perform any function. Bile salts and pancreatic lipase break down the dietary. The bile salts and the pancreatic lipase, which I mentioned, the gastric lipase, lingual lipase, all of them act on the dietary triglycerides to break them into free fatty acids, diglycerides, and also monoglycerides. And lastly, I come to the digestion of nucleic acids. The pancreas also release nucleases that also digest nucleic acid. Nucleic acids may be of two types, DNA and RNA. So the nucleases may be of two types, deoxyribonuclease and ribonuclease. Deoxyribonuclease acts on DNA, ribonuclease acts on RNA, and breaks them into the smaller nucleosides. Again, two other enzymes, that is the nucleosidase and phosphatase, act on these nucleosides to break it into pentoses, phosphates, and nitrogenous bases. So this is all about the digestion of carbohydrate, protein, fat, and nucleic acids, and also how they are digested by what chemical agents and by which mechanical processes. All aspects I discuss, and I hope that you clearly understand it, and please do make an arrangement for your notification for my next class. Thank you.